Hello, everyone, and welcome to today's webinar, No Pressure, HPLC, the analytical alternative for charged and polar analytes, presented by Dr. Vince Remcho, a professor of chemistry and materials science at Oregon State University. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by cIEF. To learn more about our sponsor, please visit them at cIEF.com. Now let's get started. I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, simply click on the Support tab found at the top right of your presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Vince Remcho, a professor of chemistry and materials science at Oregon State University. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Welcome, Dr. Remcho. You may now begin your presentation.
Great. Thanks very much, Susie. I'm grateful for the opportunity to share some thoughts and insights on the topic of doing HPLC or HPLC alternatives with no pressure. So thank you again. Let's get started. I'd like to begin by offering up some rather typical analytical lab situations that actually fit in many other parts of life as well. First one would be trying to do everything with just one technology. We tend to be driven by the technologies that are available to us in our own laboratories right now, and sometimes don't open our minds to alternatives that might fit better. So rather than one size fits all, considering a different approach to solving an analytical problem. A second typical situation might be not having enough time to do it right the first time, which forces us to do things over and over again. It's always better to try to do it the best way the first time, if that's at all possible. A third scenario is developing a transferable method, just ensuring that the method that we develop can be transferred to a QA/QC environment to maximum good effect, so that things don't have to generate a ton of questions that might come back to a method development lab and cause another iteration to have to occur. So best to develop a really truly transferable method with a small number of variables that are fixed at good values from the beginning. And then the final typical analytical lab situation might be not really factoring in the actual costs and opportunity costs to your organization that arise from not having the best alternative to an analytical technique when we launch into doing a particular kind of chemical analysis. So the way we're going to try to address these typical analytical lab situations is through some case studies. So this is called scenario-based learning, and it basically involves presenting an actual situation and then a solution or set of solutions to that particular problem. And it's a great way to learn. So we're going to look at 4 different actual situations. The first one focuses on an agricultural crop, an export from the US to Europe that was misidentified as having been contaminated with a particular pesticide. The second scenario is an actual situation again, where too much time was being eaten up in developing and running a set of analytical methods in biopharma, and it might have been easier to solve the problem using a different technology. The third scenario was a milk powder protein adulteration study, a situation that's probably familiar from the news, where an infant formula was adulterated with a different protein than was intended to have been included in the first place. And then, in the interest of time, we'll skip ahead to the next and final scenario, which is identifying a particular kind of biomolecule or bioactive molecule, an aptamer, very, very rapidly from a large mixture of candidate aptamers for a target analyte. So these will be examples. There are, of course, many other analytical challenges as well, but let's look at these four with a little bit of detail. And we'll start with U.S. agricultural export, and the particular export is walnuts, tree nuts, that were intended to have been exported from California to the EU. So in Europe, pesticide controls and pesticide regulations are different than they are in the U.S. In Europe, phosphatidylene aluminum is not an allowed pesticide. And in December of 2013, a particular walnut shipment to the EU from California was found to have a phosphatidylene residue. So it wasn't phosphatidylene aluminum that was detected, it was phosphonic acid, phosphonate, that was detected. So the conclusion was made that phosphatidylene aluminum must have been used as a pesticide on the walnuts. But in point of fact, that wasn't the case. So an analytical method that was more directly focused on analysis of phosphatidylene aluminum would have been a preferred approach. The method that was used was ion suppression chromatography. It's a fantastically useful technology, an HPLC method where we adjust the pH of the eluent to suppress the ionization of, in this instance, phosphoric acid and phosphonic acid in order to allow them to be separated chromatographically using a reverse phase method. But you notice that both of these acids, even when they're fully protonated at low pH, are still small molecules. So they're not very strongly retained on a reverse phase column when we're doing ion suppression chromatography. For that reason, we see great variability in retention time. And the chart on the left demonstrates this for phosphonate. The retention time varied over time. You see some blanks in there as well. And also for phosphate, you see variations in retention time. So this can lead to misidentification, which is not desirable. On the right-hand side, you see a calibration curve. And you notice that the calibration curve is quite linear when standards are prepared in a solvent. But when we're looking at extracts from spiked samples of various different nuts, that we don't have a very good linear response over the desired range from close to 0 parts per million to up to 20 parts per million. There's great deviation from linearity. So an alternative approach to doing this analysis, an alternative to ion suppression chromatography, would have been capillary zone electrophoresis. And what you'll notice immediately on the bottom left chart is that migration times were much more reproducible for both phosphonate and phosphate. In addition, on the right-hand side, you notice that the calibration curve is much more linear for the extract samples, for the standards that were prepared by spiking nut samples and then extracting them. The linearity was much improved over the range of concentration studied when capillary electrophoresis was the method of choice. So this allowed for more positive identification of what the contaminants were, and therefore it was possible to source those contaminants, to identify where they had come from, and therefore continue with the export. So that can result in quite a substantial savings. In addition to cost savings, we can save time in developing and running analytical methods if we are careful to select the right analytical methodology. So let's look at a different case. Here we're going to look at charge heterogeneity in biopharmaceuticals. So protein therapeutics are growing in popularity and growing in their positive effects on human and animal health. When we look at a protein therapeutic, though, there are many different kinds of concerns that we might have and many different kinds of analyses we may be interested in doing. We might be interested in looking at protein fragments. And there are many great analytical technologies for doing that, including slab gel electrophoresis, reverse phase HPLC, or potentially capillary electrophoresis. In addition, we might be interested in aggregates of proteins, or in charge variants, or in the potency of a particular protein therapeutic. Many other possibilities as well. Let's look in particular, though, at charge variants. With charge variants, the goal would be to consider as many drug candidates and manufacturing conditions as possible initially, but increase the rule-out rate so that we can narrow this down to really strong drug candidates as quickly as possible. Earlier decisions that are enabled through more comprehensive data will allow us to more rapidly bring a new product to market and prevent us from eliminating something that is a potential drug candidate, a strong drug candidate, by erroneously looking at too little data. So our goal is to develop high throughput analytical tools. So in a particular case of protein therapeutics and in particular charge variants, our alternatives of consideration might be ion exchange chromatography, capillary isoelectric focusing, and capillary zone electrophoresis. So when we talk about charge heterogeneity, what we're talking about is small variations in charge. And in this particular case, we're looking at the C-terminal lysine heterogeneity that might be possible in an antibody, a therapeutic antibody. One mechanism for looking at that particular heterogeneity is through ion exchange chromatography. Broadly utilized, very powerful technique. In this particular methodology, the positively charged protein would interact with the negatively charged packing material or sorbent, and the negatively charged species would flow through more rapidly. So there'd be a difference in retention time between the two different analytes. In developing a modern ion exchange method, we need to control the ionic strength of the buffer, the pH of the buffer that we're using as an eluent. We may be interested in running a gradient in either ionic strength or pH. We'd want to control, obviously, the flow rate and the column temperature. We have to carefully select the sorbent that we're going to use from any of a handful of different column manufacturers. So many different variables to control. So optimization, for instance, with this National Institute for Standards and Technology monoclonal antibody as a target test analyte, or just a case study analyte, we'd have many different variables to consider. Again, the salt gradient that we may use, so an ionic strength gradient or a pH gradient, and each of these is going to give us control over the analytical outcome. They will help us to optimize the separation so that we can separate acidic variants from basic variants relative to the main or target analyte, the target therapeutic that we're trying to isolate. So many different variables that would have to be optimized using careful design of experiment as we generate an ion exchange method. And this might be a typical result for this NIST monoclonal antibody. An alternative that has a different set of variables that we would need to control would be capillary isoelectric focusing. So isoelectric focusing is generally considered to be one of the, if not the, highest resolution separation techniques available in the modern market. So this is a capillary-based separation technique where we establish a pH gradient within the capillary. We would apply the sample, turn on the power supply, and analytes are separated by migrating in the direction of their isoelectric point, their PI. When they arrive at that point in the pH gradient, they essentially stop. And if they diffuse in one direction or the other, they then pick up a small amount of charge because they're now in a different pH region, and that causes them to migrate right back to their PI once again. So they're very sharply focused at their isoelectric point, the pH that corresponds to that within the pH gradient. And once again, you can see that we can get a great separation of this NIST monoclonal antibody that demonstrates that the primary component is that main peak at the center, the very tall peak that goes off scale, and it separates the acidic from the basic species once again. So a very similar outcome to what we saw with ion exchange chromatography. But once again, many different variables to consider. The ampholyte mixture, the manufacturer, or the agent that produced those ampholytes, any additives that we may need to use, for instance, a denaturant or spacers, and then the focusing time, the time we allow the experiment to evolve. So it could be important to consider a different kind of scenario for this particular separation, an alternative technology that has fewer parameters that need to be optimized, and therefore fewer places where a mistake can be made, in particular when a method is transferred from the method development lab into the QA/QC environment. The two parameters for this other method, capillary electrophoresis, would be just the buffer pH and identity, and if we chose, the addition of any zwitterionic additives. One of the great beauties of considering a capillary electrophoresis method is that a number of kits of all the reagents that are necessary are already available for a series of different kinds of analyses. This takes out much of the solution prep component of the entire separative process and allows us to rely on pre-made solutions that make the separation much, much easier to reproduce as we move through periods of great periods of time. So another benefit of looking at capillary electrophoresis is that its efficiency and resolution are nearly as good as capillary isoelectric focusing. So here, the CE separation that you see is almost as good as the isoelectric focusing separation, but there are fewer variables to control, and therefore reproducibility can be greatly improved. In fact, as we compare CE to other alternatives like isoelectric focusing and ion exchange chromatography, you'll note that it is capable of operating on a wide variety of platforms, that it has a total analysis time that is as short as or shorter than most of the alternatives. It offers excellent resolution, and it allows us to get detection limits that rival those that we're able to achieve with other techniques. So clearly, a very viable methodology. So CE has been validated across many different sites for many different applications, including a monoclonal antibody application like the one you just saw. That's true because CE has been demonstrated to be a very robust technique, a very reliable technique, and again has been employed to good effect using the same target analytes in multiple different laboratories with similar or comparable results. So let's move on to just another application example, and this would be milk powder additives or adulteration of infant formula or powdered milk through the addition of whey protein or soy protein. So it's important to be able to identify these because these are allergens potentially for many individuals. It turns out powdered milk, because it's a very complex mixture, is easily adulterated. And it can be adulterated most commonly with either whey or soy protein. Whey is just the leftover liquid from cheese production. It's a waste product that does have high protein content and therefore is quite valuable if it's not used as an adulterant and instead as an intended additive to a milk product. Soy protein could be an additive of great utility also for diluting milk or fortifying milk. But soy allergies are quite common, especially in toddlers and infants. If we contaminate a milk product with whey or soy protein, there are many different kinds of effects that could occur, including nausea, thirst, bloating, cramps, and all kinds of unhappiness for both the small child and for the parents. So identifying whether the protein content of a particular formula is as it was intended is very important. And again, because milk is very complex and difficult to analyze, we need a very high resolution analytical technique in order to do the analysis of milk. So some common analytical options for this kind of analytical issue would be gel electrophoresis or high-performance liquid chromatography. Gel electrophoresis is inexpensive, but it's relatively low resolution, has limited quantitative capability, and is very manual, very labor-intensive. HPLC, on the other hand, offers automation, very high resolution, but has the drawback of many variables to adjust, a selection of solvents, gradients, columns and column chemistries, and of course sample preparation, a very powerful technique that does require expertise. A very straightforward high-resolution alternative is capillary zone electrophoresis. And in 2018, a really informative article was published in the Journal of the Association of Official Analytical Chemists International, AOAC International, demonstrating that capillary electrophoresis is readily capable of separating low molecular weight whey protein additives into milk and whey additives of high molecular weight into milk from the casein component. In fact, the method was recognized in 2019 as the 2018 Method of the Year, capillary electrophoresis for this particular separative need. And capillary electrophoresis is now the standard for analyzing milk powder imports into Mexico. So with respect to the international market, cIEF has demonstrated its capability, its reproducibility, and its value as a good alternative to analytical technique to HPLC, ion exchange, and gel electrophoresis. Let's look at one final case study or scenario, and that is the search for an aptamer that has a high binding constant for a particular target analyte. Typically, this is a very time-consuming search. So an aptamer is an oligo, typically an oligonucleotide, but sometimes an oligopeptide, that has higher order structure that allows it to bind strongly to a target analyte. The way that we identify aptamers is by starting with a random library of oligos and screening them against the target analyte and selecting only those that have a very high binding constant for that target analyte. The beauty of aptamers is that they are non-toxic and non-immunogenic. They have very predictable pharmacokinetics. Once they've been identified, they're relatively easily prepared in bulk at relatively low cost. They have a long shelf life and are very stable. And because they are developed synthetically, they can target toxins or other analytes that don't necessarily generate an immune response and therefore aren't good candidates for the development of antibodies in an animal system. Another beauty is that they often will refold after they're exposed to denaturing conditions. This means that we can use them to bind very selectively something from solution and then purposely denature them to release what they've bound and then refold them and reuse them. They're also very useful for simple antidote generation for various different toxins. So for that reason, the identification of aptamers from these large random libraries is a very important task, although it is a difficult task. SELEX is a family of technologies that are among the most commonly used to identify high binding constant aptamers from random libraries. And two of the most popular methodologies there are the use of a nitrocellulose membrane filtration-based SELEX process, which allows us to independently separate after several selection rounds, typically up to 12, a target aptamer that has a high binding constant for our target analyte. Another possibility is the use of affinity chromatography and or magnetic bead-based SELEX. And in this instance, we would take our target analyte and immobilize it on an affinity reagent and then screen against that affinity column the entire library of aptamers. So this requires immobilizing the target analyte, and therefore it's a difficulty if the target lacks an affinity tag that allows us to easily immobilize it. So for that reason, it would be great if we could screen without having to immobilize the target analyte. And it is possible to do that. And Mike Bowser at the University of Minnesota was one of the very first to demonstrate that this is possible. So his SELEX method uses capillary electrophoresis, a high-speed technique, a high-resolution technique with a reasonable peak capacity that allows for minimal sample dilution and allows for the identification of an aptamer from an aptamer library that very selectively binds a particular target analyte. So as you see on Mike's slide, he was able to identify a number of key advantages to the cE-SELEX approach. Fewer rounds of selection are necessary. There's a lower opportunity for nonspecific interactions, few if any non-binding sequences, the ability to target small targets in free solution without having to immobilize them, and the great possibility for automation because we have CE systems that are now very well automated from a variety of vendors. So it's useful for separations of small molecules to large proteins, dramatically faster, and requires only 2 to 4 rounds of SELEX versus many more rounds in the other technologies that I just shared with you. This can save quite a lot of time. So I hope what you've seen is that capillary electrophoresis is a really powerful technique and an alternative to HPLC, but I want to speak to this idea of no pressure HPLC. In high-performance liquid chromatography, we drive our mobile phase through the system by using a pressure gradient. That generates a flow profile that distributes analyte zones more broadly than would be ideal. In a capillary electrophoresis system, be it a CZE system, an MEKC system, or even a CEC system, our flow profile is very flat. It is essentially the same flow velocity cross-sectionally across the entire capillary. That is less dispersive. It results in much sharper peaks and much higher efficiency. So CE separates based on differential migration in an electric field. It's the ratio of the charge of our analyte to its hydrodynamic volume that allows us to resolve different components from one another. The 2 most common variants of CE are capillary gel electrophoresis and capillary zone electrophoresis, but there are other alternatives that are much closer to HPLC in how they operate. But they do use the exact same instrumentation as capillary zone electrophoresis, a very simple system that's readily automated and that takes advantage of the low volume of a fused silica capillary in order to do a very high resolution separation. And these 2 techniques are Micellar electrokinetic capillary chromatography, in which we use a surfactant present in the mobile phase at a concentration above its critical micelle concentration so that we form transient micelles. These micelles form and fall apart, but they're together long enough for analytes to be able to partition into the hydrophobic core and back out. So in addition to the electrophoretic migration component that can lead to separation of charged analytes, we find that MEKC allows us to separate species that are neutral by partitioning them between 2 phases, which is a chromatographic form of separation. So here we have both an electrophoretic and a chromatographic separation occurring at the same time with no external pressure applied to the system. Another variant is capillary electrochromatography, which many groups have studied, and my group made some contributions a number of years ago to the study of capillary electrochromatography. Here, instead of using micelles to provide a sorbent for a chromatographic separation, we use a packing material, the same kinds of packing materials that we're accustomed to using in high-performance liquid chromatography, but we use electroosmosis as the motive force for bulk transport. And because we are using electroosmosis, it's a very flat flow profile, and we get all the efficiency advantages here that we would get in any other capillary electroseparation technique. So that allows us to do, once again, HPLC with no pressure as an alternative using the exact same kinds of instruments that we use for capillary electrophoresis. So there are many different applications for this family of CE techniques that include CE, MEKC, and CEC, and many other technologies as well. And these applications are in pharma and biopharma, in many different applied markets, including the ag biomarket, and many other applications that have been developed in academic laboratories that have yet to be realized in a production environment, but have the potential to be realized there. So I hope what you've seen is that it is possible to use capillary electrophoresis as an alternative to HPLC, as an alternative to ion exchange, as an alternative to gel electrophoresis, or even isoelectric focusing, and to use it to really good effect so that you can solve common analytical problems in the laboratory to much greater effect. And with that, I'll close and we'll gladly take your questions.
Thank you, Dr. Remcho, for your informative presentation. And we will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So let's get started. Looks like we have some incoming questions from our audience now. Vince, what kinds of detectors are available for capillary electrophoresis systems?
Well, Susie, I'm glad that question got passed along. The most popular option and kind of the standard for detection in a CE system is a UV-Vis detector. But photodiode array detectors are available, as are laser-induced fluorescence detectors, and even electrospray ionization interfaces to mass spectrometers are available, which adds quite a bit of additional analytical power to the CE instrument. Remember, too, you can use that CE instrument for more than just capillary zone electrophoresis. You can use it for MEKC, you can use it for CEC, you can use it for isoelectric focusing. So lots of different applications, and your choice of detector, in part, is predicated on which of those mechanisms you are most interested in using for your particular analytical problem.
Thank you, Vic. And are mass spec interfaces available for CE systems?
They are indeed. Like I said, the most popular one is the electrospray ionization interface. SELEX has a CESI system that is a very well-developed alternative within the field of possibilities, so I encourage people to consider that. But again, if we're going to interface to a mass spectrometer, we have to be careful about what mechanism of separation we're going to choose to use. If we're going to do MEKC, for instance, that requires a lot of extra care because we don't want to introduce the surfactant to the mass spectrometer. So much more popular to use the capillary zone electrophoresis mechanism when we're interfacing to a mass spec.
Thank you. And our next question from our audience member is the quantitative reproducibility of capillary electrophoresis as good as it is for HPLC?
Great question. The quantitative reproducibility of CE can be quite good due to the automation of the injection step. The qualitative reproducibility can be equally good to HPLC. One of the great strengths of liquid chromatography is that we use a 6-port sample valve with a sample loop to introduce the sample. So it's a fixed volume if we inject a full or make a full loop injection. In CE, we're using either an electrokinetic sample introduction methodology or pressure or vacuum-based injection methodology. So it's very easy to vary the volume, but reproducibility sometimes won't be quite as good quantitatively for CE as it is for HPLC. But the beauty of it is the technique is very rapid. So we get around the problem by running many replicates, several replicates, often in less time than it takes to run one sample by HPLC, and that gives us good statistical control over quantitative reproducibility.
Thank you, and thank you to our audience members for these great questions coming in. Our next question is, what steps have to be taken to ensure that the qualitative reproducibility of CE is optimized?
Well, qualitative reproducibility really has to do with retention time, or in the case of capillary zone electrophoresis, migration time reproducibility. So for that, what we need to do is make sure that we've conditioned the capillary properly before a given run. A very typical routine would be at the beginning of the analysis day to do an extensive etching and flushing of the capillary. If it's a non-modified capillary, that is. If it's a modified capillary, then there are washing procedures that are particular to that particular chemistry. But for a non-surface modified capillary, we would etch and rinse, and then run some standards, and then a mild etch and rinse, and samples, and then periodically run standards throughout the day. And if we follow that protocol, then qualitative reproducibility, migration time reproducibility becomes quite good.
Thank you, Vince. One audience member writes in, I have read about discrimination in CE sample introduction. Is this a universal problem, or is there a solution to it?
Oh, this is directly related to the previous question, so I'm glad it came along. So discrimination is the introduction of one analyte in a sample preferentially to other components of the sample. And this happens when we're making an injection with a variety of different charged analytes that have differing electrophoretic mobilities. So those with the strongest electrophoretic mobilities in the direction of the field that's being applied to the capillary, those will be introduced in the highest concentration, most rapidly. So it's basically like the start of a foot race. At the beginning of the race, those runners who are naturally the fastest will move more rapidly down the course and towards the finish line. And those of us who are a little slower will be introduced not quite as preferentially. So that kind of discrimination can be avoided if instead of an electrokinetic injection, we make a pressure or vacuum-based injection in which everything is introduced very uniformly. And with the automation in modern CE instruments, that's a very easy thing to do.
Thank you, Vince. And we have time for one more question from our audience. You mentioned heptamers can be used instead of antibodies and do induce immune response. Can they be used as vaccines?
Oh, super interesting question. Well outside of my area of expertise. I do know there's great interest in using aptamers therapeutically. But again, not being an immunologist, it's just outside of my field of study. You know, the interesting thing about aptamers from an analytical perspective is their tremendous selectivity. They really do rival antibodies in their selectivity, and because of that, they make great immunochromatographic sorbents. And so for medical diagnostic purposes, they have tremendous potential. And that makes them, once again, a really great fit for electrokinetic separations, for techniques that have an inherently high peak capacity that are good for looking at really complex samples. Great question.
And thank you, audience members, for those great questions that you brought in. And Vince, would you like to provide a closing remark for the audience before we leave today?
Absolutely. I'd like to start by just thanking SCIEX for the opportunity to share some thoughts and ideas. It's been a great pleasure to focus on some true case scenarios, some real analytical problems that have been solved using pretty innovative analytical technologies. So I'm grateful for that. But most of all, at SCIEX, I want to thank Peter Holper and Hans de Wald for their great support, and Matt Salem for his support as well. And thank you, Susie, for hosting today.
Of course. And I'd like to once again thank you for your time and your important research. I'd also like to thank LabRoots and our sponsor SCIEX for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we didn't have time for today and those submitted during on-demand registration will be addressed by Dr. Remcho via email address you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share this email with your colleagues who may have missed today's live event. You will be redirected now to the registration page of our next SCIEX webinar titled Widely Applicable Charge Variant Analysis of Therapeutic Proteins, presented by Peter Holper, Application Scientist at SCIEX. This webinar will be broadcast live on February 28th at 9 a.m. Pacific Time. We hope to see you there. Bye-bye.